All right, well, thank you very much for joining us and your interest in the Engler Lab and what we're up to. So we'll be sharing 6 or 7 projects today, telling you about what goes on in the lab. The first is a project from Alina Nelson, and I'll be sharing what she is up to and our cardiovascular interests. So she takes patient-derived induced pluripotent stem cells and converts them into ventricular cardiomyocytes, so you can see if they beat in the dish. And ultimately, we're interested in seeing how these patient mutations influence things like traction forces, as shown in the bottom left, in the center here, calcium flux, and then ultimately, in the lower right-hand image, you can see changes in their architecture. So are their sarcomeres more or less organized? So with that, we'll turn things over to Evan Tang, who is a senior graduate student in the lab, and he'll tell you about his project.
Okay, hi, I'm Evan from the Engler Lab, and I specialize in looking at how genetics influence endothelial cell susceptibility to disease. So look at this by differentiating iPSCs into endothelial cells and looking at their key characteristics. An example of this is permeability. So specifically, we made a microfluidic vessel to study this in 3D. So as you can see on the picture on the left, so these endothelial cells we cultured are able to form linear blood vessels. And there's a picture of the immunofluorescence staining that we've done. So we can also use these in vitro vessels to study how fluidic blood flow can affect these vessels' permeability and other characteristics. Yeah.
Thanks. Next on our list is postdoc Natalie Kirkland.
Hi there. So I'm using the fruit fly as a model to study aging and cardiovascular mechanobiology. Flies are rapidly aging, show a highly conserved cardiac proteome, and due to their simple architecture, allow us to conduct imaging and biophysical measurements. As you should be able to see in the movies, we can compare the physiological function of the hearts with aging, or with the power of fly genetics, compare mutants for their role in heart function. I'm currently assessing how age-associated nuclear remodeling influences heart function, and I've used methods such as genetic perturbation, AFM, and sequencing to start to address this. So if you'd like to know more or use this model to address a question of your own, you can contact Professor Engler now.
Thanks. Next on our list is graduate student Giselle Gonzalez.
Hi, so for my project, I'm working on developing a conductive polymer maturation platform for cardiomyocytes derived from stem cells. To do this, I'm using electrospinning to create an extracellular-like structure that I can then seed my cells on to determine how my stem cell-derived cardiomyocytes mature over time. A lot of the techniques I use include scanning electron microscopy to visualize my actual scaffolds, atomic force microscopy to determine the stiffnesses, as well as immunofluorescence imaging.
Thanks. Next up on our list is Ben Yeoman, a graduate student in the lab.
Hi, I'm Ben, and I'm studying how cell adhesion in the tumor microenvironment affect the biomechanics of cancer metastasis. So as a primary tumor progressives, you have a stiffening of ECM surrounding the tumor. And it's pretty widely known that cells tend to migrate up stiffness gradients. So they would durotax from the softer stroma into the tumor. So it's paradoxical how these tumor cells metastasize and migrate from this tumor into the bloodstream. So we've fabricated these photo pattern gels where we have alternating soft and stiff regions, and can see cells based on their adhesion strength and observe how they migrate between these two stiff regions, or the soft and stiff regions. So what we found is that less adherent cells are more contractile, and that leads them to become ageotactic and migrate away from these stiffer regions, which would show that they are more metastatic.
Great. Thank you so much, Ben. And our last presentation comes from postdoc Katie Birmingham.
Hi, my name is Katie, and my research focuses on determining if in vitro differences in adhesivity correlate with differential metastatic potential in vivo. So I use a parallel plate flow chamber to sort cancer cells based on their adhesive phenotype. And this allows me to collect two fractions of cells, cells that we call weakly adherent, and then cells that are strongly adherent. And then I take these fractions of cells and I inject them into mice, and I quantify the extent of tumor growth. And I'm currently in the process of doing different variations of these experiments, but our preliminary results show that cells that are weakly adherent form significantly more lung metastases than cells that are strongly adherent. And this agrees with these in vitro adhesivity differences, and we hypothesize that the weakly adherent cells have a greater migratory potential, which allows them to leave the site of that primary tumor and enter secondary tissues or organs where they can form that metastatic tumor.
Great. Well, thank you all for listening to our research today, and best of luck in your search for a PhD program and lab this year.